Let's study general virology. General characteristics of viruses. So definition, viruses are defined as acellular organisms whose genomes consist of nucleic acid but never both, either DNA or RNA, and which obligately replicate inside the host cells. So characteristics are like obligatory intracellular parasites. They possess either DNA or RNA, never both. They are filtrable, smaller than bacteria, no cellular organization, no cell wall, cell membrane, cell organelle, including ribosomes, lack enzymes necessary for protein and nucleic acid synthesis, multiply by complex process, cannot be grown on artificial cell-free culture media, can be grown on embryonated eggs, tissue and cell culture, not susceptible to antibiotics, susceptible to interferon. Morphology of viruses. Nucleic acid is the central portion of the virus. So nucleic acid may be single or double stranded, circular or linear, segmented or unsegmented. Viruses are classified based on nucleic acid as DNA or RNA viruses. Capsid, the covering of the nucleic acid is called capsid. It is made up of protein subunits, polypeptides called capsomers. The function of capsid protect the nucleic acid core from external environment. In non-enveloped viruses, initiates first step of viral replication, antigenic and specific for each virus. Envelope. So some viruses are enveloped. So envelope surrounding the nucleocapsid. It is lipoprotein in nature. Lipid part derived from the host cell membrane and protein part is virus coded. It is made up of subunits called peplomers. Peplomers project as spikes on the surface of the envelope. The significance of envelope more susceptible to heat and lipid solvents like ether. It is antigenic, facilitates entry of the virus. More than one kind of papillomas can be seen in some viruses, like influenza virus contains heme agglutinin and neuraminidase papillomas. Most viruses are enveloped, except non-enveloped DNA viruses are parvovirus, adenovirus, papovirus, and non-enveloped RNA viruses picorna, hepatitis A, and hepatitis E virus. Size of the viruses. Size vary from 20 to 300 nanometer in size. It is measured by the electron microscope based method. Smallest virus, parvovirus, 20 nanometer. Largest virus is the pox virus, 300 nanometer. Because of the small size, viruses can pass through bacterial filters and they cannot be visualized under light microscope. They are seen under electron microscope. Viral symmetry, three types: helical, example Ebola and rabies virus; icosahedral, example adenovirus and poliovirus; complex viruses, bacteriophage. Different shapes of viruses like spherical, brick shape, which is seen in pox virus; rod shaped, bullet shape, it is seen in the rabies virus; tadpole shape, it is seen in bacteriophage; filamentous, it is seen in Ebola virus. Classification of viruses. So they are according to the nucleic acid, DNA viruses and RNA viruses. So let's see DNA viruses. So first is herpes viridae. So viruses are herpes simplex virus 1, herpes simplex virus 2, varicella zoster virus, Epstein-Barr virus, cytomegalovirus, human herpes virus 6, 7, 8. Hepadnaviridae, hepatitis B virus, parvoviridae, parvovirus P19, papovaviridae, human papilloma virus HPV, JC virus and BK virus. Pox viridae, variola, that is smallpox, and molluscum contagiosum virus. Adenoviridae, human adenoviruses. So these are DNA viruses. RNA viruses, picornaviridae, poliovirus, coxsackie virus, enterovirus, rhinovirus, hepatitis A virus, calciviridae, hepatitis E virus, togaviridae, rubella virus, western equine encephalitis virus, western equine encephalitis virus, flaviviridae, yellow fever virus, dengue fever virus, and the hepatitis C virus. Coronaviridae, coronavirus, rhabdoviridae, rabies virus, filoviridae, Marburg and Ebola virus, paramyxoviridae, parainfluenza virus, mumps virus, measles virus. Orthomyxoviridae, influenza viruses ABC. Guineaviridae, hantavirus. Arenaviridae, Lassa fever virus. Reoviridae, rotavirus. Retroviridae, HIV. Human immunodeficiency virus. These are RNA viruses. Viral multiplication or replication. So stages, first stage is attachment. So viruses get attached to the host cell membrane by the receptor recognition. Then virus penetrates penetration. Then uncoating. So all the parts of the viruses get uncoated. Then biosynthesis. So there is protein synthesis by transcription, protein synthesis, and translation mechanism. Final protein synthesis, then maturation. So different parts of the viruses gets assembled and then release either by lysis of the cell, that is lytic cycle, or by budding and release, that is called lysogenic cycle. Pathogenesis of viral infection. So infection takes place on the body surface, then it causes localized infection. Example, rotaviruses, intestinal, influenza respiratory, warts on skin. Then movement of the virons, lymph node blood causes primary viremia. Then the movement to the possible sites of replication, that is blood vessel, bone marrow, liver or spleen. Then they enter blood and then causes secondary viremia and then shading blood or other organs. So shading blood, that is hepatitis B virus, lungs, salivary gland or kidneys. Example, mumps, measles, cytomegalovirus, brain, rabies, polio, measles, skin, varicella virus, nasal and oral mucosa, measles, varicella and rubella. Lab diagnosis of viral infection. So indications to start antiviral drugs, screening of blood donors for HIV, hepatitis B and hepatitis C, surveillance purpose for outbreak or epidemic investigation, and to start the post-exposure prophylaxis in AIDS or hepatitis B virus infection. So first step is sample collection. So samples like blood, throat swab, nasopharyngeal swab, swab from lesions are taken. Precautions taken. Swab collected in VTM, viral transport media. pH is maintained and organism is viable. Cold chain should be maintained. Temperature 4 to 8 degrees Celsius for 24 hours. For longer duration, minus 20 degrees Celsius freezing is done. Laboratory diagnosis of viral diseases, direct demonstration of virus by electron microscopy, immunoelectron microscopy, fluorescent microscopy, light microscopy, histological staining to demonstrate inclusion bodies, immunoperoxidase staining. Detection of viral antigens by various formats such as ELISA, direct immunofluorescence, ICT, flow-through assays. Detection of specific antibodies, conventional techniques such as HAI, neutralization test and complement fixation test. Newer diagnostic formats such as ELISA, ICT, flow-through assays. Molecular methods to detect viral genes, nucleic acid probe for detection of DNA or RNA by hybridization. PCR for DNA detection by amplification. RT-PCR for RNA detection. Real-time PCR for DNA quantification. Real-time RT-PCR for RNA quantification. Isolation of virus by animal inoculation, embryonated egg inoculation, tissue culture, organ culture, explant culture, cell line culture. In cell line, primary, secondary, continuous cell lines. Specimen collection, ideal specimen, nasopharyngeal swab or lavage fluid, nasal aspirate or to less extend throat swab. Swabs with synthetic tip, polyester or dacron swabs are best for best specimen collection. Cotton or alginate swabs are unsatisfactory. Transport swabs are immediately transported, put inside the viral transport media, kept at 4 degrees Celsius during transport up to 4 days. Thereafter, minus 70 degrees Celsius. Direct demonstration of virus. So first is electron microscopy. So we know size and shape of virus. So rabies bullet shape, rotavirus wheel shape, coronavirus penta, corona shape, papillomas, adenovirus space vehicle shape. Then immunoelectron microscopy, fluorescent microscopy, and light microscopy. So in light microscopy, demonstration of inclusion bodies, like example, rabies virus in dog is negri bodies. So inclusion body, aggregates of virons or viral proteins and other products of viral replication that alter staining property of the cell. Host cell. The characteristic of special viral infections, they have distinct size, shape, location, staining properties by which they can be demonstrated in virus infected cells under the light microscope and help in diagnosis of the viral infection. So types are three: intracytoplasmic, intranuclear, and both. So intracytoplasmic, generally acidophilic and can be seen as pink structures when stained with giemsa or using methylene blue stain. Most pox viruses and negri bodies seen in rabies virus. Intranuclear are basophilic in nature. Example, Cowdery had classified them into Cowdery type A, that is seen in herpes simplex infection. Both intracytoplasmic and intranuclear inclusion gives the appearance of owl's eye appearance seen in cytomegalovirus. What is serological method? So detection of viral antigen by various formats such as ELISA, direct immunofluorescence, immunochromatography, flow through assays. And that is seen in HIV. Detection of specific antibodies, your diagnostic formats such as ELISA, immunochromatography, flow through assays. Molecular methods related to viral genes, nucleic acid probe for detection of DNA or RNA by hybridization method, PCR for DNA detection by amplification, reverse transcriptase PCR for RNA detection, real-time PCR for DNA quantification, real-time RT-PCR for RNA quantification, that is viral load. Fifth step is the isolation of virus. So first animal inoculation, but because of the ethical issues, animals are not used. Second, embryonated egg inoculation. So hence embryonated eggs are used for cultivation of viruses. So parts of the eggs are chorioallantoic membrane for pox virus, allantoic cavity, influenza vaccine and yellow fever vaccine, amniotic cavity, isolation of influenza virus, and yolk sac, arboviruses, rickettsia, chlamydia. Third is tissue culture, organ culture. Organ can be used. Then explant culture, that is parts of the organ, and cell line culture. And types are primary, secondary, continuous. In continuous, malignant cells are used so that they divide continuously. Then the growth is seen by cytopathic effects. So these are morphological changes produced by virus in the cell line detected by light microscope. Example, syncytium or multinucleated giant cell formation seen in measles, diffuse rounding and ballooning of cells seen in HSV, herpes simplex virus. Bacteriophages, they are viruses that infect bacteria. Morphology, tadpole shape, head, tail, and tail fibers are there. Significance, phage typing in Staphylococcus aureus, phage assay, phage therapy, diagnosis, cloning vector, transduction, gene transfer in bacteria, codes for vaccines.
Hope you find this session useful for your understanding and for your exams. Thank you.